For the first time in human history, we possess the tools to modify the genetic instructions inside a living cell with pinpoint accuracy. This system is known as CRISPR Cas9. Its transformation into a programmable genome editor earned a Nobel Prize for its pioneers, including biochemist Jennifer Doudna and her collaborator Emmanuelle Charpentier, who co-discovered the specific RNA components required to direct the system to any desired location in the genome. But we didn't build this machinery from scratch. We adapted it from a survival mechanism that bacteria have used for eons. For billions of years, bacteria have been locked in a microscopic war against invading viruses called bacteriophages. Surviving an attack, the bacterium extracts a snippet of the virus's DNA as a genetic record. It integrates this viral fragment into its own genome, slotting it between identical markers of repeating code. These archived sequences are what we call CRISPR. If that virus ever returns, the bacterium uses that archived memory to produce a guide that leads a defensive enzyme straight to the invader, shredding its DNA before it can take over the cell. This evolved immune response provided the ideal foundation for scientists to engineer a tool that can be programmed to find and edit human genes. To use this in medicine, researchers utilize an engineered version of the system consisting of two parts: a custom-built guide RNA and the Cas9 protein. The guide RNA works like a GPS, scanning the 3 billion letters of the human genome. It only stops when it finds an exact match positioned next to a short specific tag called a PAM sequence. Once the guide RNA locks onto the target, it recruits the Cas9 protein, the enzyme that performs the physical work of cutting the DNA. Cas9 unzips the DNA double helix and severs both strands, creating a precise break at that exact location. But the cut itself isn't the final edit. The modification actually happens when the cell tries to repair the damage. The cell's default response to a break is to quickly glue the ends back together. This rapid repair is often imprecise, adding or removing random letters. This scrambles the instructions, silencing a disease-causing gene. However, providing a healthy DNA template during repair allows the cell to copy it, pasting a corrected sequence directly into the genome. We are seeing these mechanics work in patients with inherited blood disorders, such as sickle cell disease. Groundbreaking treatments involve extracting stem cells, editing them using CRISPR to produce healthy hemoglobin, and returning them to the patient. Clinical results show that patients who once faced a lifetime of pain and hospitalizations are now living symptom-free. We have moved from theorizing about molecular mechanics to functional cures for genetic disease. CRISPR has changed the landscape of biology because it is programmable, relatively inexpensive, and easily deployed in almost any lab. Beyond human medicine, it is being used to engineer crops that can survive a changing climate and to program immune cells that are more effective at killing tumors. But as the technology advances, we face a major ethical boundary: the difference between treating a single patient's cells and editing a human embryo. Edits made to an embryo, known as germline editing, are heritable. Any changes would be passed down to that person's children and all future generations, making them a permanent part of the human lineage. This concern became an international crisis when rogue experiments on human embryos were revealed at a global summit, highlighting the lack of uniform international regulation. There are still technical risks, such as off-target effects, where the system accidentally cuts and damages healthy, essential genes. There is also the question of equity. If these customized therapies remain incredibly expensive, they may only be accessible to the wealthy, deepening the divide in global health. Finally, we must decide where the line is drawn between curing a disease and selecting for non-medical traits, like height or athletic ability. We have learned how to write the code of life. Our challenge now is to determine exactly which stories we should tell and which ones are better left as they are.